Okay, good day students. Welcome to our lesson on the structures of proteins. So today we want to look at the primary, the secondary, the tertiary and the quaternary structure of proteins. We'll also have a look at the alpha helix and the beta pleated sheets in the secondary structures of proteins. So as you saw there are four main structures of proteins. So the first one is all just about the amino acids. Then we're looking at how the amino acids bond and then interactions between the side groups in those polypeptides, and then further additions for quaternary structure. You can see that this diagram on the right is actually more about quaternary structure here. You can see a variety of different bonds, which we'll have a look at in more detail later. So the primary structure of a protein is the number, the type, and the sequence of the amino acid subunits. So you start at the N-terminal, remember, and then you finish at the C-terminal end amino acid. Now the entire shape of the protein is determined by the precise order of amino acids, and that the protein structure is actually found experimentally, and that's through protein sequencing. And you're going to cook your proteins essentially with you'll have heat about 6 molar sulfuric acid or hydrochloric acid, I think it was, and use column chromatography. And you can also see here is an example of a protein. And this is a very simple one. This is bovine insulin. And you can see that it's got two different polypeptides in it, and then they joined here. This one has actually got a disulfide bond here. There's two sulfurs there joining it. Okay, the secondary structure of proteins. Now we'll have a look at first of all the hydrogen bonds. So between particularly what the double bond, carbon double bond to the oxygen. So the oxygen has a negative partial charge, then the hydrogen has a positive partial charge. And so you're going to get some different types of folding in your polypeptide chain. So the first one we'll look at is an alpha helix, and they'll just coil up like a spring. The other one is a beta pleated sheet, and that's where the sections also line up in parallel, forming a beta pleated sheet. Now the secondary structure is highly ordered, and it's stabilized by hydrogen bonds in particular. Now keratin has a helical structure, so it's found in hair and wool fibers. So the hydrogen on that NH group has a partial positive, and it joins with the oxygen, which has a partial negative on each of those carbon-oxygen double bonds, and that happens about every 4 amino acid subunits. Now silk is a protein with a beta pleated sheet. It's got small side groups, and they can line up closely, and that enables the hydrogen bonds to form between each of those polypeptide chains then. The tertiary structure of proteins, and this is the overall three-dimensional structure. We'll spend a little bit of time looking at this. Now the R group properties, I say like polar and non-polar, and acidic and basic, influence the shape. You can have a variety of different interactions. So hydrophobic, remember they're non-polar. So these amino acids orient themselves towards the center of the polypeptide to avoid water. The disulfide bridge, now this is particularly cysteine, forms a bond with another cysteine through its R group. That's got to be a covalent bond, hasn't it? Now hydrogen bonds over here between the hydrogen and an oxygen, for example, you'll see that happening. What else? Hydrophilic, so the amino acids, they're going to orient themselves towards the water, and ionic bonds, positively charged R groups, can also bond together. So there's quite a lot of different ways of folding. Should also mention dispersion forces as well, sorry. Yeah, so there's quite a lot of different types of bonding and folding that can occur in proteins. Proteins can be up to about 500 amino acids. Let's have a look at some of these ones here. So remember, hydrogen bonds occur between hydrogen and oxygen, nitrogen, or carbon in particular. And what do we see here? We just notice that here's our hydrogen bond forming between two different parts of a polypeptide chain. Now, dipole-dipole interactions, well, they're like hydrogen bonds, aren't they? But they're not as strong. So we've got molecules with a dipole. So, for example, between sulfur and hydrogen, oxygen, hydrogen, nitrogen, hydrogen. And so a dipole-dipole interaction linking two parts of the polypeptide chain, and we're just looking at that there. Ionic compounds or ionic interactions. So we've got, it looks like ammonia, doesn't it? And then an acetate ion joining together there. So we've got the bond there between them. And then the covalent cross-links, and this is this disulfide bridge between two cysteines. Oh yeah, we should also talk about dispersion forces, and you can see that yeah, we've got some non-polar, and so it happens in particular with the non-polar groups. And you just remember that the dispersion forces are caused from temporary dipoles, so the movement of electrons, and so that will occur more in large molecules, so they'll play an effect of attracting peptides to peptides in a protein as well. And finally, if we look at the quaternary structure of proteins, sometimes you're going to get several polypeptides joining together, or polypeptides joining with non-protein molecules. Now, hemoglobin's an interesting one. We've got 4 peptides joined together. Now, these peptides are globins, so essentially they're alpha helices. They're coiled and folded, and each of these helices has an oxygen binding site that contains iron. So this makes hemoglobin actually a lot more effective than a basic chemical interaction. It's better for transporting oxygen around your body than any other molecule of its size. So it's quite an astounding molecule. What else? We just note that the hydrophobic ends, the hemoglobin, going to be pointed towards the inside away from the blood. And then the outside, of course, is going to be what's attracted to the water in the blood. A little bit today, we've described and explained the primary, secondary, and alpha helix and beta pleated sheets, tertiary and quaternary structure of proteins. I'd like you to get on to your mastery questions when you're done, please.